Hello, I'm Dr. Mullan from Andor Technology, and I'm going to try and give some insight into how Andor's products are being used to diagnose Covid19 and research the SARS-CoV-2 virus that is causing the pandemic that we're currently dealing with. To give some background to Andor Technology, Andor Technology started off as a spin-off technology company from Queen's University Belfast, and a group of researchers there found out that they needed some kind of a very sensitive sensor to help them with their studies. And it didn't take too much time later until they found out that other scientists also needed these very high sensitivity detectors. And as a result of that, the Andor Technology company was born. And now we produce not just these highly sensitive cameras, but also systems and also the software as well to control those systems and also analyze all of that data. So we also build these cameras in Belfast, and we have engineers, R&D scientists, software engineers, and of course the sales, marketing and support teams that are all there to support those activities. Normally, our customers are involved in a diverse range of areas. One of these is astronomy, so that's looking at things like the sun, and it may seem a bit unusual, but you do actually need a very sensitive camera to detect small wavelengths of light from the sun and study it. But it's also very important for studying exoplanets, so that's planets that are outside our solar systems, and in recent years also tracking all of the space debris that is moving around our planet and can cause problems for communication satellites and so on. Another big area for us is cell biology, so that's scientists that are looking at things such as cancer research, neuroscience, so that's Alzheimer's disease, Parkinson's disease and so on. And then also the wide range of different diseases that you have, whether that's heart disease or ones that are caused by bacteria or different kinds of viruses. In terms of viruses and virology itself, for many years the Andor cameras and other products have been used to study this area. And this has been to look at these virus and cell interactions that happen. And we can actually see in this image here, we can see these bright areas in the cell in the middle, and this is showing virus replicating within the cell. So in this area then, we're trying to look at antiviral drugs to help treat those disorders, and also in vaccine development. Normally this is viruses such as HIV, influenza, hepatitis, and those other ones that would be of public health interest. This of course means that we're going to cover a wide range of different systems and setups. On the left here there is an Andor camera up on a mountainside somewhere, so there's an Andor camera mounted to this telescope at an observatory. And then on the right hand side we see quite a complex Andor imaging system which is used for some kinds of cell biology studies. So that's quite a complex setup. So there's different cameras, there's a microscope, there's an incubator to keep cells at the correct temperature, and then there's all these lasers and other components as well to support all of those imaging things that are going on. So although that's a very diverse range of areas of what people are looking at, one thing is common between them all, and that is how we use light to make these precise measurements and study small differences in things such as small molecules that are within cells, right through to faraway planets. So light has a range of properties that we can use to do such studies. So we can split light into distinct wavelength using different kinds of prisms, like here, or different kinds of filters which allow us to use different colors. And for the kind of cell biology type studies, a very important discovery was found in jellyfish that have this green fluorescence. And many years later, we have a whole range of different colors, and we can label cells and components within cells with these different types of fluorescent tags. So that means that we can study different kinds of structures and how genes are regulated within different organisms. Without these, a cell, if we put light through it, is pretty much transparent because it's mostly composed of water. So these are very powerful tools that we can use. And this image that was moving around earlier, that is a zebrafish, which we could see all the range of these different coloured fluorophores within it. So this zebrafish then is labelled with a range of different fluorescent proteins, and we can see the different structures within it. And this is where these high sensitivity cameras come in. Unlike our mobile phones and other kinds of cameras that we have, these ones are aimed at detecting the very smallest amounts of light that there is. So unlike our normal cameras that we have, these ones aren't color cameras, they are just monochrome or black and white cameras, so they will detect all of the wavelengths of light, all of the different colors of light, whereas a normal kind of camera will have different pixels within it, which will only pick up different colors of light. And that means then that these cameras can be much more sensitive. Of course, some similarities are that the sensor itself is made of the same material, and it has the role of converting the light and packets of light called photons into an electrical signal. And these cameras are quite large as well and heavy because they're cooled to low temperatures to keep the noise as low as possible. Another aspect which has become very important in more recent years is the role of software, and not only just controlling the cameras and other systems that these are connected to, but also how you make sense of all that information that you have to pick up some quite subtle differences between different conditions that you may be looking at. So in the next part, what I want to look at is some of the interesting kind of studies that people from a wide range of disciplines, so it's not just the virologists, it's astronomers, it's astrophysicists, plant scientists, neuroscientists, and many other areas. So many of these customers that we have, as the pandemic started to use their expertise and their skills from these different light microscopy and other skills to try and tackle some of these key questions that are posed as a result of the COVID-19 pandemic. The first one of these is one that we've really all at some point thought quite a lot about, and that has been, do masks make any difference in reducing the transmission of COVID-19? And of course, any respiratory viruses, whether that's influenza, ones that cause common cold, or indeed this SARS-CoV-2 virus, which causes COVID-19, they're all spread in the form of these respiratory droplets as we breathe. And this becomes very important because they found out fairly early on that people may not realize that they are affected by this, and after a certain length of time they may encounter a number of people, and of course then that continues a chain of transmission to many other people. So a group of astronomers and biophysicists down in Cork decided that this was something that they actually had the skills to look at and see, do they actually make any difference or not? And they called this project MASK, which stands for Making Aerosol Science Known. And again, this kind of seems like an unlikely group of scientists together that would be trying to tackle that issue. But again, they had the skills and the equipment to try and study this. So they were trying to look at the emission of aerosols as somebody would breathe and see whether or not the different kinds of masks, whether this be medical masks, respirators, or masks that you would make at home, whether they would make any difference at all. And to study this, they were able to use a medical dummy, which is able to breathe a vapor of aerosol out. And then they had their lasers to try and illuminate these droplets that we can see up in the top left-hand image. And then they used this Andor camera which is in the bottom left-hand corner here. And then they were able to study as the dummy breathed in and breathed out and look at the effect quite easily of seeing what happened if you had a mask on or not, and as well then about whether the mask was fitted correctly or not. So this is just a few images just from their work that we see here. And this following image here really sums things up quite well. And on the left, we can see the image of the dummy wearing a correctly fitting mask with very minimal image of any kind of aerosols, compared to on the other side, we can see without a mask, and we can see this aerosol vapor coming out. And their findings then supported the idea that by wearing the mask, you were reducing the amount of aerosols that you would produce, and thereby that would cut down the amount of transmission of COVID-19. And this is important because things on a one-to-one basis become very important when you scale that up to a population of thousands of millions of people, and then small effects like this can have a large difference. They also pointed out that simply wearing a mask in itself, sometimes it was important that you had to make sure that the mask was correctly fitted, which of course makes sense. But we never are really relying just on one study itself. And many other groups, of course, in different countries have looked at this. And again, they're using these high sensitivity cameras. This other group in the University of Edinburgh more recently have reported similar results to the group that were in Cork had presented. And they also looked at this idea of wearing masks, and by doing that, were you able to reduce physical distancing? So again, this sums up what they found, again supporting this idea that wearing masks indeed did make a difference in reducing those aerosols that would be released from somebody who may or may not know they could be infected by COVID-19. And then the physical distancing as a result can also be reduced in certain cases because you're able to wear these face coverings. So many other studies as well as this, and that gives you a weight of evidence that we can really say beyond any doubt that yes, it does make a difference. Another big question is really to do with detecting viruses, and a question on viruses themselves, about how are they alive or are they dead? Viruses are very hard to study. Not only are they very small, they can be, say, 100 times or more smaller than one of the cells in our body, but outside of a cell, for the most part, they're showing no signs of life whatsoever. So that makes them very difficult to study. So viruses, like our own cells, have some kind of genetic code in them, which in this case of this virus is called RNA. And then they also have this protective coating called a capsid. And people will remember seeing on the outside of these viruses all of these spike-like proteins that stick out from them. So there's different ways that we can detect the genetic information of them using, say, a PCR test that people will have heard about. And then there's other ways that we can pick up those other components of the virus. But simply finding the virus somewhere does not necessarily mean that it's alive. It does not mean that it's going to infect anybody. So this is very important for many reasons. And what's made more difficult in the case of this virus is very dangerous. So it can make it difficult to study because of the different kinds of lab security that you would need to have to work with it. Then, since it's transmitted in respiratory droplets, how do you accurately recreate saliva droplets rather than just water? Because the two things are quite different. And then trying to answer that question is, how do you know if that virus is just present somewhere? So it could be, say, it could be something that has been shipped overseas, and you don't know is that a risk or not. One group has been trying to study this, and this is actually a group that looks at plant microbiology. So this is like bacteria and plant interactions. So what they were able to do is they were able to use a safe model virus. So this is rather than trying to use this dangerous, potentially dangerous virus, to use a safe virus which infects bacteria. So these viruses are called bacteriophages, and that literally means bacteria eater. So that allows you then to try and study something in a more safe way. And they were able to find one of these which in its behavior and its structure is quite similar to the one that causes COVID-19. They were then able to make saliva solutions, and then they were able to spray those, and then they were able to see after a certain length of time or under different conditions or types of surfaces, and they were able to see, are they actually there or can they still infect bacteria? So they were using these high sensitivity cameras to study the droplets as they formed in surfaces, and then also to check and see if they killed the bacteria. And that's quite a well-known technique for studying viruses. So they were able to do simple studies in a practical way, which was something that in other ways it's quite difficult to study. So this and their continuing work will really help us understand how long those viruses actually do remain infectious, and we're not just picking them up in different places. So it allows you to see where do you need to take extra care and where should we not worry about it. Another big question here is about what drugs can help better manage COVID-19. So if somebody actually gets infected, how can you treat it better? And I put this up as a good example of how not to study something. This particular drug, hydroxychloroquine, we'll call it HCQ for short. It is an old malaria drug, but it also had some additional properties to do with inflammation or preventing the inflammation. So, however, Trump found out about this particular cheap, low-cost drug. He had been saying to people this is useful for treating COVID-19. So, unfortunately, at that time, the data to confirm that was sketchy at best, and that particular drug has a number of known severe side effects. So research teams that were looking at these kind of drugs that would be useful to treat COVID-19 quickly brought this in to study it as well. And the Scripps Research Institute in Florida is a center of excellence essentially for looking at these kind of inflammatory drugs and other things that we do know are very important, such as the effect of cholesterol and aging in this disease. So they added this in to see how this would actually have any effect in the lab. So it's important in developing any drugs that you don't only just do lab-based studies, but you also understand how does that drug actually work. Are there any side effects that we need to be aware of? And then also then do clinical trials to see does it in practice with real people in a population actually work. So what they were able to find is, yes, this particular drug works in the lab, like many things do, but this is at high concentrations. And whenever this was then followed up then by clinical studies, they found out that the concentrations in reality that you would be able to use it at a safe level were so low it had no effect whatsoever on the outcome of people who had COVID-19. But, however, the good news is that many other low-cost drugs are looking very promising, and in both clinical and lab-based studies they are looking to have an effect. So hopefully as time progresses we'll see the completion of those studies, and we'll end up with a lot better drugs again to manage the COVID-19 patients. The immune system is very complicated. There's a number of different systems which are there to tackle viruses. We have cells that are able to signal to other cells that they're infected to ramp up their defenses. We have a system that the cell, which the immune system can't actually see inside it, it's able to show the immune cells that are circulating around, what's actually happening inside the cells. And then there's also antibodies as well that can be produced to bind to viruses and disable them. So there's a wide range of those and many other systems even within inside the cells specifically there to stop viruses taking control of them. But viruses are very sneaky, so they can have a range of different systems that allows them to get into cells and then replicate and continue on, and it allows them to get a foothold before those systems kick in and the cells can do anything about it. So because the viruses have a very close relationship with the cells, it's very difficult to understand those relationships and what's going on. So a team at the Guttman lab over in Caltech have been using this Andor software to help try and unravel these complex interactions. And what they actually found is that this SARS-CoV-2 virus, it doesn't just have one thing that it does to try and disable the cells and allow it to replicate. It has a range of different systems, and that allows it to knock out multiple ones of these defense systems. So this is quite interesting work that they've done. This also has another very important aspect to it, is that by being infected by a virus such as this one, it can mean that it's knocking out the immune system that you have, and that can make you more susceptible to other types of infection, such as bacteria that may be able to then start getting a foothold. So any opportunistic other diseases can actually start to cause problems for you as well, and then that could be difficult then to treat. So this team used this Amaris software to look at this area, and hopefully by understanding more about those interactions with the immune system, then that allows you to develop more accurate and better effective treatments to tackle this virus. Another problem is that with some COVID-19 patients, some of ones that things turned very badly quite quickly was that our own immune system in these cases can sometimes turn against us. And what that means is that our own immune system is essentially overreacting and causing a lot of inflammation and damage. So whenever that happens in the lung tissue, it can cause cells in there to die, and then it becomes very difficult to breathe, and you end up with potentially respiratory failure. So sometimes this effect is called this cytokine storm. But the immune system is highly complex, and it's still something that we're really trying to get to better grips with today. So Key Peng is someone who is trying to tackle this for COVID-19. And they use this Andor Dragonfly system to study the inflammatory pathway to find out what is it within this system that is actually going wrong, what's happening with COVID-19, why is the immune system behaving in this way. So this Dragonfly product is quite different from other microscope systems that could be used, because it allows you to use a range of different techniques to study cells, and you can study different types of cells very quickly, so you can get a lot of information using a range of different techniques, and then you can actually, you have a lot of data to work with and find out what's happening. So what he was able, and his team was able to do, was that they were able to find this key controller molecule that's involved in this inflammatory response of what's being switched on or disabled whenever their COVID-19 patient has been infected. And by pinpointing the exact part of that pathway, this means that you can work out what is going to be a more effective treatment than the currently available steroids and other types of anti-inflammatory drugs that are currently used, because any drugs that you use, of course, will have different side effects to them, and by trying to pinpoint something more exactly, you can really focus in on trying to help patients as quickly as possible. So part of this work was made possible by using this Dragonfly system. One thing that has come up quite often recently is to do with all these variants of COVID-19 or SARS-CoV-2, we should say. So mutations are something that are inherent or normal to viruses. They happen all the time. Influenza virus is one example that mutates rather a lot, and that's why we have to change the vaccine for it most years and adjust it. Other viruses also change as well. And in the case of this virus, in recent years, with the advances that we have in high-speed gene sequencers, it means that we can gather a lot of information about what exactly is happening across the world with these types of viruses. And these high-speed gene sequencers, again, are using the principle of light and high-speed imaging detectors or cameras, and they're able to quickly analyze the different viruses and see what are the differences between them. And then you can work out then what are the key differences from all of the ones that there are, and see which ones of those are actually significant. So this isn't just something that's done with SARS-CoV-2, that's just something that we're all focused on. This is also done for HIV, Ebola, influenza virus, mumps virus, all those other viruses that have public health concerns. So by having these high-speed gene sequencers, you're able to keep track of that. So each of the different parts of the code of this RNA for these viruses, all these different little codes that we have here, you see G, C, A, T, that are all given different colors. And by having these high-speed cameras and being able to pick up those different colors that are emitted, you're able to scan through the sequence that has been labeled with all these little different tags, and then you're able to see whereabouts those changes have happened to the viruses. So this is very, very useful that we're able to do this, because 15 years ago, 10 years ago, we wouldn't have been able to do this, and we wouldn't really have been seeing any of this information. The last thing that I was wanting to look at was some future technologies, just some other interesting things that there are. One of these is an interesting thing called Pac-Man. So again, with many of these different things, they try and give them some kind of interesting acronym or name. And what this particular one does is, bring that back up again. What this particular one does is that this uses a gene editing technology. So the gene editing technology that it uses is called CRISPR. So this is one that the Nobel Prize for Chemistry was awarded for last year because it's so important. And this CRISPR is like a kind of a set of scissors, and it is able to go in and make precise cuts in the genetic code of whether it's a virus or our own DNA, wherever we want to cut the genetic code, you're able to do it with this. And this system was originally found in bacteria. And again, this was a bacterial defense mechanism to protect them against these bacteriophage viruses that we mentioned earlier. So they found that these bacteria had some system that was chopping up these viruses. So that kind of technology can be useful for gene editing, which will happen at some point in the future. But it's also useful for having some kind of a system to target viruses and to be able to go in there like Pac-Man and essentially bind onto those viruses and chop them up. So that's quite an interesting thing, but it will not appear probably for some time. And some other things that people are looking at are antibody treatments. If you give somebody a vaccine, it takes a number of weeks, as we all know, to build up levels of your own antibodies and to be able to have some resistance to that particular virus. So some people aren't able to respond to vaccines. Their immune system may not work correctly. So that's for people that have, for example, you could have people that are undergoing cancer treatment. You could have people that have had organ transplants, as well as other people as well. The immune system doesn't respond to vaccines. They can have allergies as well for some different types of vaccines with the different things that may be in them. And this means then that vaccine is not going to be suitable for them, so they will not benefit directly from the vaccine. They will be relying on other people to have the vaccine, and then hopefully then they should not be exposed to people then that are carrying something that may cause harmful effects to them. So antibody treatments then are another way of providing people antibodies to a virus that you're wanting protection from, and you can use it for potentially for people that don't have their own system to tackle the virus, but it also allows you to give something to people and allows them to have faster response. So a number of our customers are looking at this area as well, which hopefully in the next months we will see some interesting things coming through to do with that area. The last one I have in this slide is to do with creating more robust vaccines. Although drugs like the Pfizer vaccine that we will have, or vaccines like the Pfizer one that people will know of, is a modern technology vaccine and has a lot of benefits to it. You will be aware that it has a cold chain to it, that you need to make sure that it's kept at low temperatures during transport, and then also slightly lower temperatures, but still within refrigeration for you to administer it. So the problem with that is that for some countries that have high temperatures, less well-developed medical systems and services, that's not very practical for them to administer. So, and in order to tackle these kind of viruses that cause pandemics, you really need to be able to treat people in those areas. So some researchers are looking at other ways of delivering vaccines that are much more stable at higher temperatures. So that's another thing that will appear at some point, which is quite an interesting thing to happen. So these things that we've just covered are just really small parts in a jigsaw, and by adding into all the other research that is being done, this allows you to have a much fuller picture of the COVID-19 pandemic situation. And by building up that better picture, then it allows you to have a better understanding of all of the aspects of that virus. And then hopefully that allows you to move on in an informed way to better manage reducing how it's transmitted, and then if anybody is infected by it, how you actually treat that disease so it becomes much more manageable and it gets us out of eventually of where we currently are today. So if anybody wants to find out anything more about Andor Technology or some of the things that our customers are working on well beyond COVID-19, so all of the other interesting research, be that more cell biology related or whether that being physics or astronomy related, please check out our Andor website or also our social media pages as well. So we always try and keep people informed about what our different customers are doing. So thank you very much.